Hi, my name is Mathieu Quesnel-Vallieres. I am a postdoc in the Barash, Lynch, and Thomas Tikkunenko labs at the University of Pennsylvania. The work that I'm presenting today was conducted in close collaboration with Anupama Jha, a former grad student in the Barash lab. I'm happy to report our results on a project which aimed to draft a molecular definition of cancer by interpreting deep learning models. We already have a pretty good definition for the genetic components of cancer. If we look at the Cancer Genome Atlas data, we know that 89% of samples have at least one driving mutation in one of the 9 cellular pathways that are shown here. And these could be defined as the core cancer cellular pathways. Now, the question that we had was, do these mutations converge on transcriptomic variations? And also, are there additional non-genetic determinants of cancer? We trained feed-forward neural networks with transcriptomic data in the form of protein coding gene expression, long non-coding RNA gene expression, or splice junction usage, which we derived from a data set that we assembled from 14 RNA-seq data sets, including TCGA and GTEx, and additional smaller data sets, together comprising over 7,000 healthy samples and over 4,000 cancer samples, covering a large array of tissue and tumor types. We trained the neural networks to distinguish between healthy tissues and cancer. And once we had successful models, we interpreted these models to derive attribution values that would help us figure out which transcriptomic features were most important for our models to distinguish between healthy tissues and cancer. We first use protein coding gene expression to train a model that was very good at predicting which sample is cancer and which sample is healthy, as shown here by this high level of precision and recall. And what's very important, we also found that the model was very accurate across different cancer data sets. And this shows that our model can predict samples on the basis of their biological signature and not because of confounding factors. Interestingly, we were also able to train successful models using long non-coding RNA gene expression and splice junction usage, which is remarkable because on top of having a protein coding gene expression signature of cancer, this shows that there exists a robust long non-coding RNA gene expression signature and splice junction signature. Once we had trained successful models, we wanted to know which were the genes and the splice junctions that defined these models. What we found is that there is very little overlap between our definition of cancer based on transcriptomic features and known genomic features of cancer. What we see here is the overlap between genes that we found to be overexpressed in cancer in red, or expressed at lower levels in cancer in blue here, and the overlap with known tumor suppressor genes in green, or known oncogenes. And you can see that this overlap is very minimal, meaning that we find genes that were not previously associated with cancer by genomic studies. What about the function of these genes? What this enrichment map of Go terms show is that the nodes that are in blue correspond to protein coding genes that have lower expression in cancer. And these genes are enriched in functions that are traditionally associated with cancer development, including histone modification, chromatin regulation, transcription. On the other hand, we have a very distinct separate network of nodes composed of terms that are enriched amongst genes that have higher expression in cancer or that have splice junctions enriched in cancer. And that these functions are have a lot to do with our RNA processing. So we have RNA splicing. We also have non-coding RNA processing or ribosomal RNA involved in these functions. So not only are the genes that we find to be enriched in cancer different from the ones that were previously genetically associated with cancer, they also assume very different functions. Next, we did some functional analyses to assess whether the genes that we identified in our models might be contributing to disease onset or progression. We first looked at the evolutionary conservation amongst protein coding genes that are enriched or depleted in cancer relative to a neutral set, which we defined from genes that have attribution values close to 0. We also found that genes that are enriched or depleted in cancer have higher selective pressure against loss of function relative to the neutral set. Finally, I want to highlight how the genes that we find to be highly expressed in cancer or have splice junctions enriched in cancer and that have roles related to RNA processing are highly interconnected with the core cellular pathways of cancer, which I mentioned at the beginning of the introduction. So in all these colors here, this suggests that RNA processing might be a process that is downstream of the previously identified cellular pathways that contribute to cancer. In summary, we drafted a transcriptomic definition of cancer, which differs from the known genomic definition of cancer. Genes with lower expression in cancer are associated with a traditional view of cancer, while genes with higher expression or with splice junctions enriched in cancer have functions related to RNA processing. We found a high conservation and selective pressure against loss of functions in genes with high attribution values, which suggests that these genes have essential functions in cancer cells. And finally, we showed that deregulation of RNA processing genes and aberrant splicing are pervasive features across a large array of solid tumor types. I thank my mentors, Dr. Brash, Dr. Lynch, and Dr. Thomas Tikhonenko for their support. And I thank my colleague, Dr. Anupama Jha, for her important role in this research.